Ready? Okay. So thank you very much for inviting us here. Today I wanted to talk to you a little bit about advanced biofabrication technologies to be able to generate functional tissues in the lab. Specifically about 3D bioprinting and bioreactors for tissue engineering applications. And I'm going to do it from the point of view of my company. I work at Regema 3D as a product specialist, and we specialize in the development of custom biofabrication solutions. So I'm going to tell you a little bit about what we're doing, how we're doing it, and the applications that this technology might have, because the main focus of the technology is, of course, the creation of functional tissues in the lab, but it also has many other applications, as you will see. So first, a little bit about our company. So we're called Regema 3D. We're based in the south of Spain, in Granada. And our main mission is the development of innovative biofabrication technology, custom biofabrication solutions for the fields of bioprinting and regenerative medicine. And ultimately, to be able to develop solutions that improve the patient's quality of life. We were founded in 2015. And right now, this year is our 10th anniversary. We are present in more than 30 countries, and we collaborate with more than 200 research groups that work in many different applications. So what do we do? What do we offer? Our main specialty, our main focus is in 3D bioprinting technologies and the development of bioreactors. But obviously we are involved in all the steps of the biofabrication process. So we also distribute biomaterials for these technologies, right? We also are about to start distributing cellular components as well. So the main two components needed to generate 3D bioprinted tissues, the cells, the biomaterials. We obviously design and develop 3D bioprinters, and we are developing now a line of bioreactors to functionalize 3D bioprinted tissues. We can do all of this thanks of our infrastructure. So we are very focused on customization. We are the only company, We are the only company in the world with the capacity of customizing this biofabrication solution, this biofabrication hardware. And we do that thanks to our dedicated engineering department that has the capacity to custom make, to develop ad hoc solutions for particular research groups, particular projects. That's actually the way in which we have developed most of the tools that we have right now in the catalog. So most of them we have developed specifically for a particular project, and later on they were added to our catalog. We're also very focused in innovation. That's why we also have a dedicated R&D department and biofabrication and cell culture laboratory, where we develop both internal projects and also collaboration research projects with other institutions and private companies. We also perform bioprinting tests, biocompatibility tests for our collaborators and partners. And our main focus, our goal in the long term, would be the clinical translation of our technologies. We are developing our products. Right now, we are focused on research, but our main goal is to actually bring these technologies into clinics, right? That's what we want, and that's how we can make actually an impact in society. So this is something that you probably know, but talking about how to create functional living tissues in the lab, the main strategy that's currently being tackled, it's the generation of 3D cell cultures, 3D biomimetic constructs using 3D bioprinting, right? And for that, we need several ingredients. We need a scaffold that we can make using compatible biomaterials, such as PCL, PLA, but also collagen, GELMA. We have many different options to adapt our scaffold to the particular tissue that we want to generate. Then we can create a cell-laden hydrogel combining the biomaterials with the cell type that's going to be used depending on the tissue we're working in. And with that, we can generate, as I said, a biomimetic construct. But this is just the beginning, right? 3D bioprinting allows us to create 3D cells, 3D structures that mimic the structure and composition of in vivo tissues. But if we want to have a living functional tissue, we also need to replicate function. So not only structure, but function. So 3D bioprinting allows us to replicate structure and composition, but how do we replicate function? Well, we can do that by allowing these biomimetic constructs to maturate, and we can do that in bioreactors for tissue engineering. So bioreactors for tissue engineering are systems that allows us to replicate relevant physiological environment depending on the tissue, right? So that allows us to control temperature, to control pH, CO2, etc. But not only that, because that's the same thing that an incubator does, right? What's different is that these bioreactors are active environments. They have the capacity to apply a stimuli depending on the tissue that are going to promote this maturation. So, for example, the bioreactor you can see here is the first bioreactor that we have developed and commercialized. It's a bioreactor for application in osteochondral regeneration, which I'm going to explain a little bit more later. So I've discussed two steps in the biofabrication process, right? The 3D bioprinting to replicate structure and bioreactors to replicate function. So now discussing a little bit more about 3D bioprinters. These are the two systems that we currently have in our catalog. So these both bioprinters are really solid systems, and they have a characteristic that's the most important for me, and I'm going to repeat myself because they are custom systems. But this is very important because every tissue has different needs, different requirements, and you cannot achieve that with the only system, with only one system, right? You have to be able to either get different systems or adapt your system to be able to tackle different applications. And that's what we do. The fact that we have a catalog of different components, so different print heads, different printing surfaces that you can combine to generate different configuration to adapt specifically the bioprinter to your application, right? That also gives you the capacity to actually adapt the system, for example, for a project that's evolving and its needs are changing, or to have one system for a multidisciplinary group, right, that have different needs. And not only that, but we also have, as I said, the capacity to custom design specific tools that might cover different needs than the ones that we can offer right now in our current catalog. So our systems are also something very important about our systems. They're mechanical extrusion based. So within extrusion bioprinting, you have different options. You have pneumatical based system and mechanical based system. So mechanical systems, such as ours, have many advantages toward pneumatic extrusion. The main one being the fact that with mechanical extrusion, you can extrude but also retract the material, which we cannot make with pneumatic extrusion. So that gives you the capacity to actually control the deposition of the material in a finer manner, resulting in better resolution and better shape fidelity. Also, it gives you the capacity to work with both high viscosity materials, but also low viscosity materials or bioinks that require gentle pressures. And as important as the hardware is, it's also important the software, right? The software of the bioprinter will limited what you can and cannot do with the system. That's why we also develop our own software, and actually we're just about to release a new advanced slicer software that gives you a lot of functionalities and allows you to design very complex structure, to the point that you can actually configure different groups, different regions of one same scaffold with different configurations. So you can have a scaffold that has an inner mesh that's different in different regions of the scaffold, actually mimicking heterogeneous structure that you can find in the human body. So after discussing a little bit about 3D bioprinting, I want to just mention some of the applications of the technology. So obviously, tissue engineering, being able to produce tissue models to research different biological phenomenon in these tissues. Obviously, regenerative medicine, disease models, right? For example, for drug testing, microfluidics as well. Even though 3D bioprinting technology haven't been developed specifically for microfluidics, it actually can be used for this application as well. Drug development, for example, to develop personalized drug forms that can be 3D bioprinted in the point of care. Drug testing, as I mentioned, and also biomaterial development for biomaterials in 3D bioprinting technologies. And why not food printing, right? There's always new applications being discovered. So just as an example of some of these applications, I'm showing you here some papers, some publications from collaborators. In this paper, for example, researchers developed a malignant melanoma model. So actually a 3-laminar model with different composition and different cell type for to mimic the different 3 layers of the skin. And this is a very interesting paper because besides developing this model, then they characterize it mechanically and biologically to make sure it was the closest to real melanoma, malignant melanoma. And they also tested with different anti-tumor drugs. They screened it for different anti-tumor drugs. And finally, they implanted in an animal model to see if this model was able to generate tumorigenesis in the animal model, and they saw that in fact it did. So it's a very complete paper, and you can see how this tissue model can be used for drug screening and for other different applications. This is another example of a publication. In this case, the application would be the development of a drug delivery system. This drug delivery system was developed for clonic applications, and in this case the researchers you cannot see the whole structure here of the scaffold, but you can see it has a peculiar structure with different circumferences. They actually tried different structures of the scaffold, and their goal was to achieve the best kinetic release for this particular drug. This is another paper where one of our collaborators actually developed a cardiac patch with contractile capacity. So the goal is to research the development of these cardiac patches with applications in cardiac regeneration. And they actually achieved not only to develop a cardiac patch with contractile capacity, but also they saw the formation of endothelial networks in the patches. In this other paper, the researchers developed a skin model. In this case, a healthy skin model for drug testing, pharma testing, I meant cosmetic testing, etc. And this model was also a successful 3-laminar model. We also have some collaborators working in hospitals, in research units in hospital. For example, we have here an example where these researchers developed 3D printed enterical mesh with bacterial capacity. So you can see there are many different applications for 3D bioprinting only, but this is just the first step. As I said, the second step would be the maturation of these 3D bioprinted scaffolds in bioreactors. So as an example of this technology, I want to introduce you BIMAMNI, which is the first bioreactor that we have developed for mitochondrial applications. So the BIMAMNI bioreactor has two main different parts. The first one would be the movement simulator, so all the mechanics that allow to actually mimic the movements you will find in a human knee joint. And the second part would be the biocapsule. So this is the most important part. So in the biocapsule, you can find a human knee joint model, 3D printed knee joint model, right? That's able to, when combined with the movement simulator, replicate this same mechanical stimuli you will find in a human knee joint. And the model has two particularities. So in the model, you can see that in the femur condyles, you can find an area that's the lesion area, right? This area allows introducing a 3D bioprinted, yes, bioprinted this time, 3D bioprinted scaffold, biomimicking cartilage tissue. So you can 3D bioprint your scaffold, mimicking the lesion area, and then introduce it in the bioreactor where it's gonna receive these mechanical stimuli, promoting maturation of your 3D bioprinted cartilage construct. The biocapsule also contains sensors that monitor all the different parameters that you need to maintain to have this physiological relevant environment. So temperature, pH, CO2, and also load. So here you can see here closer this biocapsule and the sensor unit, and also the femur condyles with the lesion area. So this would be the default model, right? But what's interesting about this bioreactor is that the knee joint model is made out of medical information from a patient, in this case a healthy patient, but it can be personalized with medical information from real patients, including the dimensions of the lesion area. Therefore, you can actually print your knee joint model, right, personalized for your patient, 3D bioprint the scaffold, and then introduce it in the bioreactor for functionalization, for personalized functionalization of the scaffold. So again, the software, which is also very important, in the case of the bioreactor, the software actually controls temperature and pH and monitors, as I said, temperature, CO2, pH, and average load during time. This is a bioreactor that is made for long-term experiments. And then the most important part would be the experimental design. So the bioreactor allows you to select different movements, up to 4 different movements: traction, compression, flex, extension, rotation, and lateral movement. You can select different movements to make a sequence of movements, right? That's gonna be your experimental design. So this sequence will mimic the movements in a human knee joint. And obviously you can select also the time that each movement is gonna be displayed and the cycles per minute of each movement. And you can also introduce pauses, for example, for exchanging of media, or just to mimic, let's say, a resting period. So here are some examples, some initial results of the validation testing that we've doing with bioreactor. So you can see how different parameters are staying perfectly constant during the whole experiment. Here is just to say, this slide is just to say that obviously you can actually design your scaffolds with different inner mesh, with different structure to modify their mechanical properties, you know, to better fit the requirements of your research. And finally, we have an image of a live death assay where we can see that cells are perfectly happy in the bioreactor, that they are able to grow and to differentiate And this is part also of the validation assay of the bioreactor. And finally, so to sum up, what the BMAGNI bioreactor offers is the possibility of monitoring and controlling temperature, pH, CO2 during scaffold maturation to study the influence of a scaffold structure, porosity, and mechanical properties on cell differentiation, and to evaluate the effects of mechanical stimuli on chondrogenic differentiation. So this sums up in a better understanding of the influence of physiological-like stimulation on chondrogenic differentiation. So this is obviously focused towards cartilage, but we are actually developing other bioreactors. We are involved in several collaboration projects where we are developing other tissue-specific bioreactors as well. So, for example, we are involved in NeuroFab project, where we are developing a bioreactor for electrical stimuli of biomimetic nervous tissue 3D bioprinted scaffolds. In this same project, we also have developed a new bioink based on GELMA and functionalized with graphene fibers that is electroconductive. We also working in the BioLast project, where we are developing a bioreactor to functionalize tendon and muscle. And we also are working in these two projects, Reboard and Inject. So actually these two projects are the ones that are most advanced. So I'm gonna talk to you a little bit about them. So the Reboard project has a goal of developing a piezoelectric multifunctional patch able to release therapeutic factors into infarcted tissue to promote its regeneration, right? And then the Inject project aims to integrate inkjet printing technology into 3D bioprinting to bring this technology into the biomedical field by developing several pilot lines. So the development of 3D bioprinted cartilage scaffold for regeneration of osteochondral defects, but using Inject technology, the development of Inject sensors for multi-analysis of biomarkers, and the development of transdermal drug delivery systems for personalized medicine. So this is just some examples of some of the projects that we are currently involved in. And this is just to say that for us, being involved in research, other than being hardware producers, which we are, but being involved in research is very important for us, because that gives us the possibility to validate our systems for different applications, to develop new technology, and mostly to be in touch with research, to be in touch with researchers' needs, and to be able to improve and advance our technology for our final goal, which will be the translation of this technology into the clinics. This is an image of our biofabrication and cell culture lab, but we are actually based on a bioengineering hub. So we are very close from the Faculty of Medicine of University of Granada and several research institutions like GENIO or CBM that have, top-notch facilities, and it allows us, through our collaborations with them, to have access to these facilities as well. So thank you very much for your attention, and if you have any question, now would be the moment. Thank you.